अभी हम लोग क्या करेंगे, एनसीईआरटी का रीडिंग ख़त्म करके हम लोग चैप्टर का स्टार्ट करेंगे, वो भी स्टार्टिंग से। तो आज हम लोग स्टार्ट करेंगे चैप्टर 5 क्लास 12 मोलिकुलर बेसिस ऑफ इनहेरिटेंस। ओके, लेट्स रीड इट आउट। In the previous chapter, you have learned that the inheritance patterns and genetic basis of such patterns. At the time of Mendel, nature of those factors regulating the pattern of inheritance was not clear. Over the next 100 years, the nature was of the putative genetic material was investigated, culminating in the realization that DNA, deoxyribonucleic acid, is the genetic material. At least for the majority of organisms in class 11th, you have learned that the nucleic acid are polymers of nucleotides. Deoxyribonucleic acid means DNA and the ribonucleic acid RNA are the two types of nucleic acid found in living system. DNA act as a genetic material in most of the organism. RNA, though it also act as a genetic material in some viruses, mostly functions as a messenger. RNA has additional role as well. It functions as adapter, structural, and in some cases as a catalytic molecule. In class 11, you have already learned that structures of nucleotides and the way these monomers units are linked to form nucleic acid polymers. In this chapter, we are going to discuss the structures of DNA, its replication, the process of making RNA from DNA transcription, the genetic code that determines the sequence of amino acid in proteins, the process of protein synthesis, translation, and the elementary basis of their regulation. The determination of complete nucleotide sequence of human genome during last decade has set in a new era of genomics. In the last section, the essential of human genome sequencing and its sequences will also be discussed. Let us begin our discussion by first understanding that the structures of the most interesting molecules in the living system, that is DNA, and in subsequent section we will understand that why it is the most abundant genetic material and what its relationship with RNA. The DNA is a long polymer of deoxyribonucleotides. The length of DNA is usually defined as number of nucleotides or a pair of nucleotides referred to as base pair present in it. This also is the characteristic of an organism. For example, a bacteriophage known as 5174 has 5386 nucleotides. Bacteriophage lambda has 48,502 base pairs. Escherichia coli has 4.6 into 10 to the power 6 base pair, and haploid content of human DNA is 3.3 into 10 to the power 9 base pairs. Let us discuss the structure of such a polymer, such a long polymer. Structures of polynucleotide chains. Let us recapitulate the chemical structure of polynucleotide chain DNA or RNA. A nucleotide has three components: a nitrogenous base, a pentose sugar, ribose in case of RNA, and deoxyribose for DNA, and a phosphate group. There are two types of nitrogenous base: purines, adenine and guanine, and pyrimidines, cytosine, uracil, and thymine. Cytosine is common for both DNA and RNA, and thymine is present in DNA. Uracil is present in RNA at the place of thymine. Nitrogenous base is linked to OH of 1 prime carbon pentose sugar through N-glycosidic linkage to form a nucleoside. Such as adenosine or deoxyadenosine, guanosine or deoxyguanosine, cytidine or deoxycytidine, or uridine or deoxythymidine. When a phosphate group is linked to OH of fifth carbon of a nucleosides, fifth position carbon of a nucleosides through phosphoester linkage, or corresponding nucleotides or deoxynucleotides depending upon the type of sugar present is formed. The nucleotides are linked through 3 to 5 phosphoester linkage to form a dinucleotide. More nucleotides can be joined in such a manner to form a polynucleotide chain. A polymer thus formed has at one end and a free phosphate moiety at 5 prime end of sugar, which is referred to as 5 prime end of the polynucleotide chain. Similarly, at the other end of the polymer, the sugar has free OH of 3 prime carbon group, which is referred to as 3 prime end of the polynucleotide chain. The backbone of the polynucleotide chain is formed due to sugar and phosphate. The nitrogenous base linked to sugar moiety project from backbone. In RNA, every nucleotide residue has an additional OH group present in second position of the ribose. Also in RNA, the uracil found at the place of thymine, 5-methyl uracil, another chemical name for thymine. DNA has an acidic substance present in nucleus was the first identified by Friedrich Miescher in 1869. He named it as nuclein. However, due to technical limitation in isolating such a long polymer intact, the elucidation of structures of DNA remained elusive for a very long period of time. It was only in 1953 that the James Watson Francis Crick, based on the X-ray diffraction data produced by Murray Wilkins and Rosalind Franklin, proposed a very simple but famous double helix model for the structures of DNA. One of the hallmarks of their proposition was base pairing between the two strands of polynucleotide chain. However, this proposition was based on the observation of Evin Chargaff that for a double-stranded DNA, and the ratios between adenine, thymine, and guanine and cytosine are constant for equals 1. The base pairing confers a very unique property to the polynucleotide chains. They are said to be complementary to each other, and therefore, if the sequence of the base pair in one strand is known, then the sequence of other strand can be predicted. Also, if the each strand of a DNA act as a template for synthesis of new strand, the double stranded DNA thus produced would be identical to the parental DNA molecule. Because of this, the genetic implication of the structures of DNA become very clear. The silent feature of the double helix structures DNA are as follows. It is made up of polynucleotide chains where the backbone is constituted by sugar phosphate and the base project inside. The two chains have anti-parallel polarity. It means if one chain has the polarity 5' to 3', the other has to 3' to 5'. The base pair in two strands are paired through hydrogen bond, forming base pair. Adenine forms two hydrogen bonds with thymine from opposite strand and vice versa. Similarly, guanine is bonded with cytosine with 3 hydrogen bond. Here 2 hydrogen bond and here 3 hydrogen bond between guanine and cytosine. As a result, always a purine comes opposite to pyrimidine. This generates approximately uniform distance between the two strands of helix. The two chains are coiled in a right-handed fashion. The pitch of the helix is 3.4 nanometer. A nanometer is 1 billionth of a meter, that is 10 to the power minus 9 millimeter. And there are roughly 10 base pair in each turn. Consequently, the distance between a base pair in a helix is approximately 0.34 nanometer. The plane of one base pair stacks over the other in double helix. This, in addition to H bond, confers stability of helical structures. The proposition of double helix structures of DNA and its simplicity in explaining the genetic implication become revolutionary. Very soon, Francis Crick proposed the central dogma in molecular biology, which stated that genetic information flows from DNA to RNA and RNA to protein. That is central dogma. In some viruses, the flow of information is in reverse direction, that is, from RNA to DNA. Packaging of DNA helix. Taken to distance between two consecutive base pairs as 0.3 nanometer, if the length of the DNA double helix is typically mammalian cell is calculated by calculating multiplying the total number of base pair with distance, that is 6.6 into 10 to the power minus 10 to the power 9 base pair into 0.34 into 10 to the power 9 meter per base pair. It comes out approximately 2.2 meter. A length that is far greater than the dimension of typical nucleus 10 to the power minus 6 meter. How is such a polymer packaged in a cell? That's a very big question. If the length of the Escherichia coli DNA is 1.3 millimeter, can you calculate the number of base pair in Escherichia coli? Yes, of course, by dividing, multiplying. Okay. In prokaryotes such as Escherichia coli, though they do not have defined nucleus, the DNA is not scattered throughout the cell. DNA, being negatively charged, is held with some proteins that have positive charge in a region termed as nucleoid. The DNA in a nucleoid is organized in a large loop held by protein. In eukaryotes, this organization is no much more complex. There is a set of positively charged basic proteins called histones. A protein acquires a charge of depending upon the abundance of amino acid residue with the charged side chains. Histones are rich in the basic amino acid residue lysine and arginine. Basic amino acid. Both the amino acid residue carry positive charge in their inside chains. Histones are the organized to form unit of 8 molecules, that is histone octamer. The negatively charged DNA is wrapped around the positively charged histone octamer to form structures called nucleosome. A typical nucleosome contains 200 base pair of DNA helix. Nucleosome constitutes the repeating unit of structures in nucleus called chromatin. Thread-like strand bodies seen in nucleus. The nucleosome in chromatin are seen beads on string structures when viewed under the electron microscope. Theoretically, how many such beads do you imagine at present in mammalian cell? Okay. The beads on string structures in chromatin is packaged to form chromatin fibers that are further coiled and condensed at metaphase stage of cell division to form chromosomes. The packaging of chromatin at higher level requires additional set of proteins that collectively are referred to as non-histone octamer chromosomal proteins (NHC). In typical nucleus, some regions of chromatins are loosely packed and referred to as euchromatin, and the chromatin that is more densely packed and strands are dark as heterochromatin. Euchromatin is said to be transcriptionally active chromatin, whereas heterochromatin is inactive. The search for genetic material. Even though discovery of nuclein by Miescher and the proposition of principle of inheritance by Mendel were almost at the same time, but that the DNA act as a genetic material took long to be discovered and proven. By 1926, the quest to determine the mechanism of genetic inheritance had reached the molecular level. Previous discoveries by Gregor Mendel, Walter Sutton, Thomas Hunt Morgan, and numerous other scientists had narrowed the search to the chromosomes located in the nucleus of the most cells, but the questions of what molecule was actually the genetic material no one has answered. Transforming principle. In 1928, Frederick Griffith, 1928, in a series of experiments with Streptococcus pneumoniae, bacterium responsible for pneumonia, witnessed a miraculous transformation in bacteria. During the course of his experiment, a living organism has changed in physical form. When Streptococcus pneumoniae bacteria are grown on a culture plate, some produce smoothy shiny colonies represented by S, while others produce rough colonies R. This is because the S strain have a mucus polysaccharide coat, while R strain does not. Mice infected with S strain virulent die from the pneumonia infection, but mice infected with R strain do not develop pneumonia. Griffith was able to kill bacteria by heating them. He observed that the heat-killed S strain bacteria injected into mice did not kill them. When he heat-killed live injected into mice, but mice die. How this happened? Mixture of heat-killed and mice died. Moreover, he recovered living S bacteria from the dead mice. He concluded that the R strain bacteria had somehow been transformed by the heat-killed S strain bacteria. Some transforming principle transferred from the heat-killed S strain has enabled the R strain to synthesize a smooth polysaccharide coat and become virulent. This must be due to transfer of genetic material. However, the biochemical nature of the genetic material was not defined from his experiment. Biochemical characterization of transforming principle. Prior to the work of Oswald Avery, Colin MacLeod, Maclean McCarthy, 1932 to 44, the genetic material was thought to be a protein. They worked to determine the biochemical nature of transforming principle in Griffith experiment. They purified biochemicals, proteins, DNA, RNA, etc. from the heat-killed S cells to see which one could transform live R cells into the S cells. They discovered that DNA alone from the S bacteria caused R bacteria to become transformed. They also discovered that protein digesting enzymes and RNA digesting enzymes did not affect transformation, so the transforming substance was not a protein or RNA. Digestion with DNAs did inhibit transformation, suggesting that the DNA caused that the transformation. They concluded that DNA is the hereditary material, but not all biologists were convinced. The genetic material is DNA. The unequivocal proof of DNA is the genetic material came from the experiment of Hershey and Chase, 1952. They worked with virus that infect bacteria called bacteriophage. The bacteriophage attached to the bacteria is the genetic material that enters the bacterial cell. The bacteria cell treat the viral genetic material as if it was its own and subsequently manufacture more virus particles. Hershey Chase worked to discover whether it was protein or DNA from the virus that enters the bacteria. They grew some virus on a medium that contained radioactive phosphorus and some others on medium that contains radioactive sulfur. Viruses grow in the presence of radioactive phosphorus, contain radioactive DNA but not radioactive protein, because the DNA contains radioactive phosphorus but proteins does not. Similarly, the viruses grow on radioactive sulfur, contain radioactive protein, not radioactive DNA, because DNA does not contain sulfur. Radioactive phages were allowed to attach Escherichia coli bacteria. Then, as the infection proceeded, the viral coats were removed from the bacteria by agitating them in a blender. The virus particles were separated from the bacteria by spinning them in a centrifuge. Bacteria which was infected with virus that had radioactive DNA were radioactive, indicating that DNA was the material that passed from the virus to the bacteria. Bacteria that were infected with virus that had radioactive proteins were not radioactive. This indicates that the protein did not enter the bacteria from the viruses. DNA is therefore the genetic material that is passed from the virus to bacteria. Properties of genetic material DNA versus RNA. From the foregoing discussion, it is clear that the debate between the proteins versus DNA as the genetic material was unequivocally resolved from the Hershey Chase experiment. It became an established fact that it is DNA that acts as genetic material. However, it subsequently became clear that in some viruses, RNA is the genetic material. For example, tobacco mosaic virus, QB bacteriophage, etc. Answer to some of the questions such as why DNA predominant genetic material, whereas RNA perform dynamic function, messenger and adapter, whereas. So the answer for the questions, molecules that can act as a genetic material must fulfill the following criteria, which was followed by DNA. It should be able to generate its replica. It should be stable chemically and structurally. It should provide the scope for slow changes, mutation that are required for evolution. It should be express itself in the form of Mendelian characters. If one examines each requirement by one by one, because of the rules based pairing of and complementarity, both are nucleic acid. DNA and RNA have the ability to direct their duplication. The other molecules in living systems such as proteins fail to fulfill first criteria itself. The genetic material should be stable enough to change, not to change with the different stage of life cycle as with the change in physiology of the organism. Stability as one of the properties of genetic material was very evident in Griffith's transforming principle itself. That heat which killed the bacteria at least did not destroy some of the properties of genetic material. This now can easily be explained in light of the DNA, that two strands being complementary, if separated by heating, come together when appropriate conditions are provided. Further, 2' OH group present in at every nucleotide in RNA is a reactive group and makes RNA labile and easily degradable. RNA is also now known to be catalytic, hence reactive. Therefore, DNA chemically is less reactive, structurally more stable when compared to RNA. Therefore, among the two nucleic acid, the DNA is better genetic material. In fact, the presence of thymine and the presence of uracil also confers additional stability of DNA that you will study in higher classes. Both the DNA and RNA are able to mutate. In fact, RNA being unstable mutate at faster rate. Consequently, viruses have RNA genome and have shorter lifespan, mutate and evolve faster. RNA can directly code for synthesis of protein, hence can easily express the characters. DNA, however, is dependent on RNA for synthesis of protein. The protein synthesizing machinery has evolved around RNA. The above discussion indicate that both RNA and DNA can function as genetic material, but DNA is being more stable is preferred for a storage of genetic information. For the transmission of genetic information, RNA is better. RNA world. From foregoing discussion, an immediate question become evident: which is the first genetic material? It shall be discussed in details in chapter on chemical evolution, but briefly we shall highlight some of the facts or points in this chapter here. RNA was the first genetic material. There is now enough evidence to suggest that essential life processes such as metabolism, translation, splicing, etc. evolved around RNA. RNA used to act as a genetic material as well as catalyst. There are some important biochemical reactions in living systems that are catalyzed by RNA catalyst and not by protein enzyme, but RNA being a catalyst was reactive and hence unstable. Therefore, DNA has evolved from RNA with chemical modification that makes it more stable. DNA being double-stranded and having complementary strand further resist changes by evolving a process of repair. Replication. While proposing the double helical structure of DNA, Watson and Crick had immediately proposed a scheme for replication of DNA. To quote their original statement, that is as follows: "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible coping mechanism for genetic material." Watson and Crick, 1953. The scheme suggested that the two strands would separate and act as a template for synthesis of new complementary strand. After the completion of replication, each DNA molecules would have one parental and one newly synthesized strand. This scheme was termed as semi-conservative DNA replication. The experimental proof: it is now proven that DNA replicates semi-conservatively. It was shown first in the Escherichia coli and subsequently in higher organisms such as plants and human cells. Matthew Michelson and Franklin Stahl performed the following experiment in 1958. They grew Escherichia coli in a medium containing NH4Cl, heavy isotopes of nitrogen, as the only nitrogen source for many generations. The result was that N15 was the incorporated to newly synthesized DNA. These heavy DNA molecules could be distinguished from normal DNA by centrifugation in a cesium chloride density gradient. Please note that N15 is not a radioactive isotope and it can be separated from N14 only based on densities. They transformed the cells into a medium with normal NH4Cl and took samples at various definite time interval as the cells multiplied, and the extracted the DNA that remained as double-stranded helices. The various samples were separated independently on cesium chloride gradients to measure the densities of DNA. Thus, the DNA that was extracted from the culture one generation after the transform from N15 to N14 medium, after 20 minutes Escherichia divides 20 minutes, had a hybrid or intermediate density. DNA extracted from the culture after the another generation, that is after 14 minutes, second generation, was composed of equal amounts of this hybrid DNA and light DNA. If Escherichia coli were allowed to grow for 80 minutes, then what would be the proportion of light and hybrid densities DNA molecules? Very similar experiments involving use of radioactive thymidine to detect distribution of newly synthesized DNA in a chromosome was performed by Vicia Fava Beans by Taylor and colleagues in 1958. The experiments proved that the DNA in chromosomes also replicates semi-conservatively. The machinery and enzymes in living cells such as Escherichia coli, the process of replication requires a set of catalyst enzymes. The main enzymes is referred to as DNA-dependent DNA polymerase, since it uses a DNA template to catalyze the polymerization of deoxynucleotides. These enzymes are highly efficient enzymes as they have to catalyze polymerization of a large number of nucleotides in a very short time. Escherichia coli, that has only 4.6 into 10 to the power 6 base pair, compared it with human whose diploid content is 6.6 into 10 to the power 9 base pair, completes the process of replication within 18 minutes. That means the average rate of polymerization has to be approximately 2000 base pair per second. Not only to do these polymerases have to be fast, but they have also to catalyze the reaction with high degree of accuracy. Any mistake during the replication would result into mutation. Furthermore, energetically, replication is a very expensive process. Deoxyribonucleoside triphosphate serve dual purpose. In addition to acting as substrates, they provide energy for polymerization reaction. The two terminal phosphate in a deoxynucleoside triphosphate and are high energy phosphate, same as in case of ATP. In addition to DNA dependent DNA polymerase, many additional enzymes are required to complete the process of replication with high degree of accuracy. For long DNA molecules, since the two strands of the DNA cannot separate it in entire length due to very high energy requirement, the replication occurs within a small opening of the DNA helix, referred to as replication fork. The DNA dependent DNA polymerase has catalyzed polymerization only in one direction, that is 5' to 3'. This creates some additional complication at this replication fork. Consequently, on the strand, the replication is continuous, while on the other it is discontinuous. The discontinuously synthesized fragments are later joined by the enzyme DNA ligase. The DNA polymerases, on the other hand, cannot initiate the process of replication. Also, the replication does not initiate randomly at any places in DNA. There is a definite region in Escherichia coli DNA where the replication originates. Such regions are termed as origin of replication. It is because of the requirement of the origin of the replication that a piece of DNA, if needed to be propagated during recombinant DNA procedures, requires a vector. The vectors provide the origin of replication. Further, not every details of replication is understood well. In eukaryotes, the replication of DNA takes place at S phase of the cell cycle. The replication of DNA and cell division cell cycle should be highly coordinated. A failure in cell division after DNA replication results into polyploidy, a chromosomal anomaly. You will learn the detailed nature of origin and processes occurring at these sites in higher classes. Okay. Transcription. The process of copying genetic information from one state of DNA into RNA is termed as transcription. Here also the principle of complementary governs the process of transcription, except the adenosine complementary now form base pair with the uracil instead of thymine. However, unlike in the process of replication, which once set in, the total DNA of an organism get duplicated. In transcriptional, only a segment of DNA and only one of the strand is copied into RNA. This necessitates defining the boundaries that would demarcate the region of the strand of the DNA that would be transcribed. Why the both of the strands are not copied during transcription has the simple answer. First, if both strands act as a template, they would could for both RNA molecules with different sequences. And in turn, if they code for protein, the sequence amino acid in the proteins would be different. Hence, one segment of the DNA would be coding for two different proteins, and this would be complicate the genetic information transfer machinery. Second, the two RNA molecules, if produced simultaneously, would be complementary to each other, hence would form a double-stranded RNA. This would prevent RNA from being translated into proteins, and exercise of transcription would become a futile one. Transcription unit. A transcription unit in DNA is defined primarily by the three regions in the DNA: a promoter, a structural gene, terminator. There is a convention in defining the two strands of the DNA in the structural gene of the transcription unit, since the two strands have opposite polarity, and the DNA dependent RNA polymerase also catalyzes the polymerization in only one direction, that is 5' to 3'. The strand that has the polarity 3' to 5' act as a template and also referred to as template strand. The other strand, which has the polarity 5' to 3' and the sequence same as RNA except thymine at the place of the uracil, is displaced during the transcription. Strangely, the strand which does not code for anything is referred to as coding strand. All the reference point while defining in a transcription unit is made with the coding strand. To explain the point, a hypothetical sequence from a transcription unit is represented below: 3' to 5', template strand, 5' to 3', coding strand. The promoter and the terminator flank the structural gene in a transcription unit. The promoter is said to be located towards 5' end of the structural gene with respect to the polarity of the coding strand. If the DNA sequence that provides binding site for the RNA polymerase, it is presence of the promoter in a transcription unit, and also defines the templates and coding strands by the switching its position with terminator. The definition of the coding and template strands could be reversed. The terminator is located towards 3' end downstream of the coding strand and usually defines the end of the process of the transcription. There are additional regulatory sequence that may be present further upstream or downstream to the promoter. Some of the properties of this sequence shall be discussed while dealing with the regulation of one gene expression. The transcription unit and the gene. A gene is defined as the functional unit of inheritance. There is no ambiguity that the genes are located on the DNA. It is difficult to literally define a gene in terms of DNA sequence. The DNA sequence coding for tRNA or rRNA molecules also define a gene. However, by defining a cistron as a segment of DNA coding for a polypeptide, the structural gene in a transcription unit could be set as monocistronic or polycistronic. Mostly in bacteria or prokaryotes. In eukaryotes, the monocistronic structural genes have interrupted coding sequence. The genes in eukaryotes are split. The coding sequence or expressed sequence are defined as exons. Exons are said to be those sequences that appear in mature or processed RNA. The exons are interrupted by introns. Introns or intervening sequence do not appear in mature or processed RNA. The split genes arrangement further complicates the definition of a gene in terms of a DNA segment. Inheritance of a characters is also affected by promoter and regulatory sequence of a structural gene. Hence, sometimes the regulatory sequence are loosely defined as regulatory genes, even though the sequence do not code for any RNA or protein. Types of RNA and the process of transcription. In bacteria, there are three major types of RNA: mRNA, tRNA, rRNA. All the three RNA needed to synthesize a protein in a cell. The mRNA provides the templates, tRNA brings amino acid and reads the genetic code, rRNA plays a structural and catalytic role during translation. There is a single DNA dependent RNA polymerase that catalyzes transcription for all types of RNA in bacteria. RNA polymerase binds to promoter and initiates transcription. Initiation: it uses nucleosides triphosphate as substrate. Three process: initiation, elongation, termination. And polymerization is a template dependent fashion of following the rule of the complementarity. It somehow also facilitates opening of the helix and continuous elongation. Only a short stretch of RNA remains bound to the enzymes. Once the polymerase reaches the terminator region, the nascent RNA falls off. So also the RNA polymerases. This results in termination of the transcription. An intervening question is that how is the RNA polymerase able to catalyze all three steps, which are initiation, elongation, and termination? The RNA polymerase only capable of catalyzing the process of elongation. It associates with transiently with the initiator factor and termination factor. Initiate and terminate the transcription respectively. Association with these factors alter the specificity of the RNA polymerase to either initiate or terminate. In bacteria, since the mRNA does not require any processing to become active, and also since transcription and translation takes place in the same compartment, no separation of cytosol and nucleus. In bacteria, many times the translation can begin much before the mRNA is fully transcribed. Consequently, the transcription and translation can be coupled in bacteria. In eukaryotes, there are two additional complexities. There are at least three RNA polymerases in nucleus to the RNA polymerase found in the organelles. There is a clear cut division of labor. The RNA polymerase 1 transcribes RNAs 28S, 18S, and 5.8S, whereas the RNA polymerase 3 is responsible for transcription tRNA, 5S RNA, snRNA, small nuclear RNA, that is M. The polymerase 2 transcribes precursor of mRNA, heterogeneous nucleus RNA, hnRNA, precursor of mRNA. The second complexity is the primary transcripts contain both the exons and introns and are non-functional. Hence, it is subjected to a process called splicing, where the introns are removed and the exons are joined in a defined order, and hnRNA undergoes additional processing called as capping and tailing. In capping, an unusual nucleotide, methylguanosine triphosphate, is added to the 5' end of the hnRNA. In tailing, adenylate residue 200 to 300 are added to the 3' end in template independent manner. It is the fully processed hnRNA, now called mRNA, that is transported out of the nucleus of the translation for translation. The significance of such complexities is now beginning to be understood The split gene arrangement represent probably an exceed features of the genomes. The presence of the introns are reminiscent of antiquity, and the process of splicing represents the dominance of RNA world. In the recent times, the understanding of RNA and RNA-dependent processes in the living system has assumed more importance. Genetic code. During replication transcription, the nucleic acid was copied to form another nucleic acid, hence the process are easy to complete conceptualize on the basis of the complementarity. The process of translation surprise transfer of genetic information from a polymer of nucleotides to synthesize a polymer of amino acid. Neither does any complementary exist between the nucleotides and amino acid, nor could be any drawn theoretically. There existed ample evidence, though, to support the notion that change in the nucleic acid genetic material were responsible for changes in amino acid in proteins. This led to the proposition of genetic code that could direct the sequence of the amino acid during the synthesis of proteins. If determining the biochemical nature of the genetic material and the structure of DNA was very exciting. In proposition, the deciphering of the genetic code were most challenging. In very true sense, it is required involvement of the scientist from several disciplines: physicist, organic chemist, biochemist, geneticist. It was George Gamow, a physicist, who argued that since there are only 4 bases, if they have code for 20 amino acids, the code should constitute a combination of bases. He suggested that in order to code for all the 20 amino acids, the code should be made up of 3 nucleotides. This was very bold proposition because of permutation combination with 4 raised to power 3 would generate 64 codons, generating many more codons that required. Providing proof that codon was a triplet was more daunting task. The chemical method developed by Hargobind Khurana was instrumental in synthesizing RNA molecules with defined combination of bases. Marshall Nirenberg's cells-free system for protein synthesis finally helped the code to decipher. Severo-Okow enzyme was also helpful in polymerizing RNA with the defined sequence in a template-independent manner. Finally, a checkerboard for genetic code was prepared with a given table. The silent feature of genetic code are as follows. The codon is triplet: 61 code for amino acid, 3 codons do not code for any amino acid, hence they function as stop codons. Some amino acids are coded by more than 1 codon, hence the code is degenerate. The codon is read in mRNA and in contiguous fashion. There are no punctuation. The code is nearly universal. For example, the bacteria to human UUU would code for phenylalanine, and some exceptions to this rule have been found in mitochondrial codons and in some protozoans. AUG has double function. It codes for methionine and it also acts as initiator codon. UAA, UAG, UGA are stop terminator codon. It following is the sequence of the nucleotides in mRNA. Predict the sequence of the amino acid coded by it. It takes help of the checkboard. Now try the opposite. Mutation and genetic code. The relationship between the genes and the DNA are the best understood by mutation studies. You have a study about mutation and effect in chapter 4. Effects of large deletion and rearrangement of sequence of DNA are easy to comprehend. It may result in loss or gain of the genes or as a function. So as a function, the effect of point mutation will be explained here. A classical example of point mutation is challenge of single base pair in the gene for beta globin chain, and that results in the challenging for amino acid residue glutamate for valine. It results in a diseased condition that called as sickle cell anemia. Effect of point mutation that insert or deletion of base pair in each gene can be better understood by following examples. Consider a statement of the each 3 letters. Ram has a red cap, and if we insert letter B between has and red, the rearrangement of the statement would be Ram has Bray DCA cap. So similarly, if we now inserted 2 letters at the same places, that could be like this. The conclusion from the above exercise is very obvious. Insertion or deletion of 1 or 2 bases changing the reading frame from the point in insertion or deletion. However, such mutation is referred to as frame shift insertion or deletion mutation. Insertion or deletion of the thread or its multiple base insert or deletion one or multiple codon, hence one or multiple amino acid, and reading frame remains unaltered from the point outwards. tRNA, the adapter molecules. From the very beginning of the preposition of the code, it was clear to Francis Crick that 3 has to be a mechanism to read the code and also to link it with the amino acid. Because of amino acids have no structural specialties to read the code uniquely, he postulated the presence of the adapter molecules that would one hand read the code or on the other hand would bind to a specific amino acids. The tRNA, then called sRNA, soluble RNA, was known before the genetic code was postulated. However, its role as an adapter molecules was assigned much later. tRNA has an anticodon loop that has the bases complementary to the code, and it also has an amino acid acceptor end to which it binds to amino acids. tRNA are specific for each amino acid. For initiation, there is another specific tRNA that is referred to as initiator tRNA. There are no tRNAs for stop codons. The secondary structures of tRNA has depicted that looks like a clover leaf. In actual structures, the tRNA is a compact molecule which looks like inverted L. Translation. Translation refers to the process of polymerization of amino acid to form a polypeptide. The order of sequence of amino acid are defined by the sequence of bases in the mRNA. The amino acids are joined by bond which known as peptide bond. Formation of a peptide bonds requires energy. Therefore, in the first phase itself, amino acids are activated in the presence of ATP and linked to their conjugate tRNA. A process commonly called as charging of tRNA. Amino acylation of tRNA has to be more specific. If two such charged tRNAs are brought close enough, the formation of peptide bonds between them would be favored energetically. The presence of a catalyst would enhance the rate of peptide bond formation. The cellular factor is responsible for synthesizing proteins. If the ribosomes consist of a structural RNA and about 18 different proteins. In its inactive state, it exists as two subunits, a large subunit and a small subunit. When the small subunit encounters the mRNA, the process of translation of the mRNA to protein begins. There are two sites in the large subunits for subsequent amino acids to bind and thus be close enough to each other for the formation for a peptide bond. The ribosomes also act as a catalyst for the formation of peptide bond. The translational unit in mRNA is the sequence of RNA that is flanked by the start codon and the stop codon code for the polypeptide. And mRNA also has some additional sequence that are not translated and referred to as untranslated region UTR. The UTR present at the 5' end and 3' end. They are required for efficient translation process. For initiation, the ribosomes binds to the mRNA and the start codon AUG is recognized only by the initiator tRNA. The ribosomes proceeds to the elongation phase of the protein synthesizing during the stage. Complexes composed of an amino acid linked to tRNAs sequentially bind to the appropriate codon in mRNA by forming complementary base pairs with the tRNA anticodon. The ribosomes moves from the codon to codon along the mRNA. Amino acids are added one by one, translated into polypeptide sequence dictated by DNA and represented by mRNA. At the end, a release factor binds to the stop codon, terminating translation and releasing the complete polypeptide from the ribosome. Regulation of the gene expression. Regulation of the gene expression refers to a very broad term that may occur at various levels. Considering that the gene expression results in the formation of the polypeptide, it can be regulated at several levels. In eukaryotes, the regulation could be exerted at transcriptional level, processing level, transport of mRNA from nucleus to the cytoplasm, translation level. The genes in all cells are expressed to form a particular function or a The genes in all cells are expressed to perform a particular function or a set of functions. For example, if an enzyme called beta-galactosidase and synthesized by Escherichia coli. It is used to catalyze the hydrolysis of and disaccharide lactose into galactose and glucose. The bacterium use them as a source of sugar. Hence, if the bacteria do not have lactose around them to be utilized for energy source, they would no longer require to synthesize or the enzyme beta galactosidase. Therefore, in simple terms, it is the metabolic, physiological, or environmental condition that regulate the expression of gene. The development and differentiation of embryo into adult organism are also a result of the coordinated regulation of expression of several set of genes. In prokaryotes, control of the rates of the transcriptional initiation is the predominant site for the control of gene expression. In a transcription unit, the activity of RNA polymerase at a given promoter is in turn regulated by interaction with accessory protein, which affect its ability to recognize start sites. The regulatory sites can act both positively and negatively. The accessibility of promoter regions of the prokaryotic DNA is in many cases regulated by the interaction of proteins with sequences termed operators. The operator region is adjacent to promoter element in most operons, and in most cases the sequence of the operator bind as repressor protein. Each operand has its specific operator and specific repressor. For example, leg operator is present only in leg operon, and it interacts specifically with leg or repressor only. The leg operon. The elucidation of the leg operon was also result of close association between the geneticist Francois Jacob and biochemist Jacob Monod. They were the first to elucidate transcriptionally regulated system. In leg operon, polycystonic structural gene is regulated by a common promoter and regulatory genes. Such arrangements is very common in bacteria and is referred to as operon. To name few such examples, leg operon, TRP operon, ERA operon, His operon, Wall operon, etc. The leg operon consists of one regulatory gene, that is I gene. Here the term I does not refer to as inducer, rather it is derived from the word inhibitor, and three structural genes Z, Y, and A. The I genes codes for the repressor, and the leg operon Z genes codes for the beta galactoside, and which primarily responsible for the hydrolysis of the disaccharides lactose into its monomeric units galactose and glucose. The Y genes codes for the permease, which increase permeability of the cell to the beta galactosidase. The A gene encodes a transacetylase. Hence, all the three genes products in leg operon are required for the metabolism of lactose. In most of other operons as well, the genes present in the operon are needed together to function in the same or related metabolic pathway. Lactose is the substrate for the enzyme beta galactosidase, and it regulates switching on or off of the operon. Hence, it is termed as inducer. In the absence of a preferred carbon source such as glucose, if lactose is provided in growth medium of the bacteria, the lactose is transported into the cells through the action of permeases. Of the expression of leg operon has to be present in the cell all the time. Otherwise, lactose cannot enter the cells. The lactose then induces the operon in the following manner. The repressor of the operon is synthesized from gene I, and the repressor proteins bind to the operator region of the operon and prevents RNA polymerases from transcribing the operon. In the presence of an inducer such as lactose or allolactose, the repressor is inactivated by interaction with the inducer. This allows RNA polymerase access to the promoter and transcription proceeds. Essentially, regulation of leg operon can also be visualized as regulation of enzyme synthesized by its substrate. Regulation of leg operon by repressor is referred to as negative regulation. Leg operon is under control of positive regulation as well as, but it is beyond the scope of discussion at this level. Human Genome Project. In the preceding section, you have learned that it is sequence of bases in DNA that determines the genetic information of given organism. In other words, genetic makeup of organism or an individual lies in the DNA sequence. If the two individuals differ, then their DNA sequence should also be different. At least at some places, these assumptions led to the quest of finding out the complete DNA sequence of the human genome. With the establishment of the genetic engineering techniques, where it was possible to isolate and clone any pieces of DNA, and availability of the simple and fast techniques for determining DNA sequence, a very ambitious project of the sequencing human genome was launched in year 1919. Human Genome Project was called a mega project. You can imagine the magnitude of the requirement for the project if we simply define the aim of the project as follow. Human genome is said to be approximately 3 into 10 to the power 9 base pair, and if the cost of sequencing require US $3 per base pair, the total estimated cost of the project would be approximately 9 billion US dollar. Further, it obtained the sequence were to be stored in a type form in books, and if each page of the books contain 1000 letters and each book contained 1000 pages, then 33 such books would be required to store the information sequence from a single human cell. The enormous amount of data expected to be generated also necessitated to the use of high speed computational devices for data storage and retrieval and analysis. Human Genome Project was closely associated with the rapid development of a new area in biology called bioinformatics. Goals of HGP: identify all the approximately 20 to 25,000 genes in human DNA, determine the sequence of 3 billion chemical base pairs that make up human DNA, store this information in database, improve tools for data analysis, transfer related technologies to other sectors such as industries, address the ethical, legal, and social issues that may arise from the project. And the Human Genome Project was a 13 years project coordinated by US Department of Energy and National Institute of Health. During the early years of HGP, the Wellcome Trust UK became a major partner. Additional contribution came from Japan, France, Germany, China, and others. The project was completed in 2003. Knowledge about the effects of the DNA variation in individuals can lead to revolutionary new ways to diagnose, treat, and someday prevent the thousands of disorders that affect human beings. Besides providing clues to the understanding human biology, learning about the non-human organisms' DNA sequence can lead to an understanding of their natural capabilities that can be applied towards solving challenges in healthcare, agriculture, every production, environmental remediation, many more human model organisms such as bacteria, yeast, Xenohabiditis elegans, a free-living non-pathogenic nematode, Drosophila, fruit fly, plant, rice, and Arabidopsis, etc. have also been sequenced. Methodologies: the method involving the two major approaches. One approach focused on identifying all the genes that are expressed as RNA, referred to as express sequence tag, and the other took the blind approach, simply sequencing the whole set of genomes that contain all the coding and non-coding sequence, and later assigning different regions in the sequence with function sequence annotation. For sequencing, the total DNA from a cell is isolated and converted into random fragments relatively smaller size and cloned suitable host using a specialized vectors. Cloning resulted into amplification of each pieces of DNA fragment so that it subsequently could be sequenced with ease. The commonly used hosts were bacteria and yeast, and the vectors were called BAC, bacterial artificial chromosomes, and YAC, yeast artificial chromosomes. The fragments were sequenced using automated DNA sequence that worked on the principle of method developed by Frederick Sanger. Sanger is credited for developing method for a determination of amino acid sequences in proteins. These sequences were then arranged based on the some overlapping regions present in them. This requires generation of overlapping fragments for sequencing. Alignment of these sequences were human not possible. Therefore, specialized computer-based programs were developed. These sequences were subsequently annotated, were assigned to each chromosomes. The sequence of chromosomes 1 was completed only in May 2006. This was led to the 24 human chromosomes, 22 autosomes, and XY to be sequenced. Another challenging task were assigning the genetic and the physical maps on the genome. This was generated using information on the polymorphism of restriction in the nucleus recognition sites and some repetitive DNA sequence known as microsatellites. One of the applications of polymorphism in repetitive DNA sequence shall be explained in next section of DNA fingerprinting. Now the silent feature of the human genome. Some of the silent observations drawn from Human Genome Project are as follow. The human genomes contain 3164.7 million base pairs. The average genome consists 3000 bases but size vary greatly, with the largest known human genes being dystrophin at 2.4 billion bases. The total number of genes is estimated at 30,000, much lower than the previous estimated 18,000 to 1,14,000 genes. Almost all 99.9% nucleotide bases are exactly same in all people. The functions are known for over 50% of the discovered genes, less than 2% of the genome codes for protein. Repeated sequences make up very large portion of the human genome. Repetitive sequences are a stage of DNA sequence that are repeated many times, sometimes 100 to 1000 times. They are thought to have no direct coding function, but they shed light on chromosomes structures, dynamic and evolution. Chromosomes 1 has most genes 2968 and the Y has the fewest 2231. Scientists have identified about 1.4 million locations where single basis DNA differences (SNP single nucleotide polymorphisms) occur in human. This information promises to revolutionize the process to finding chromosomal location for disease-associated sequence and tracing human history. Application and future challenges. Deriving meaningful knowledge from the DNA sequence will define research through the coming decades, leading to our understanding of the biological system. This enormous task will require the expertise and creativity of the tens of the thousands of the scientists have from varied disciplines in both the public and the private sectors worldwide. One of the greatest impact of the having the HG sequence may well be enabling the radically new approach of the to the biological research. In the past, researchers study one or few genes at a time with the whole genome sequences and the new high throughput technologies. We can approach questions systematically and on a much broader scale. They can study all the genes in a genome, for example, all the transcripts in a particular tissue or organ or tumor, and how tens of the thousands of the genes and proteins work together in interconnected networks or orchestrate of the chemistry of life. DNA fingerprinting, as stated in the preceding section, 99.9% of the base sequence among humans is the same, assuming human genomes is 3 into 10 to the power 9 base pair. In how many bases sequence there will be difference. It is this difference in the sequence of DNA which makes every individual unique in their phenotypic appearance. If one aims to find out genetic differences between the two individuals or among the individuals of a population, sequencing the DNA every time would be a daunting and expensive task. Imagine trying to compare two sets of 3 to 10 to the power 6 base pair. DNA fingerprinting is a very quick way to compare the DNA sequence of any two individuals. DNA fingerprinting involves identifying differences in some specific regions in a DNA sequence called repetitive DNA. Because in this sequence a small stretch of DNA is repeated many times, these repetitive DNAs are separated from bulk genomic DNA as different peaks. During density gradient centrifugation, the bulk DNA form a major peak and the other small peaks are referred to as satellite DNA. Depending on the base composition, length of the segment and the number of repetitive units, and the satellite DNA is classified into many categories such as microsatellites, mini satellites, etc. These sequences normally do not code for any protein, but they form a large portion of human genome. The sequence show a high degree of polymorphism and form the basis of DNA fingerprinting. Since the DNA from the every tissue such as blood, hair follicle, skin, bone, saliva, sperm, etc. from the individual show the same degree of polymorphism, they become very useful identification tool in forensic application. Further, as the polymorphisms are inheritable from parents to children, DNA fingerprinting is the basis of paternity testing in case of disputes. As polymorphism in DNA sequence is the basis of genetic mapping of human genome as well as DNA fingerprinting, it is essential that we understand that what DNA polymorphism means in simple terms. Polymorphism variation at genetic level arises due to mutation, as affected you have already studied in chapter 4 and the preceding chapter in this chapter. Now, new mutation may arise in an individual either in somatic cells or in the germ cells, the cells that generates gametes in sexually reproducing organism. If a germ cells mutation does not seriously impair individual's ability to have offspring who can transmit the mutation, it can spread to the other members of the population. Allelic sequence variation has traditionally been described as a DNA polymorphism if more than one variant at a locus occurs in human population with a frequency greater than 0.01. In simple terms, if an inheritable mutation is observed in a population at higher frequency, it is referred to as DNA polymorphism. The probability of such variation to be observed in non-coding DNA sequence would be higher, as mutation in this sequence may not have any immediate impact on individual's reproductive ability. These mutations keep on accumulating generation after generation and form one of the basis of variability, polymorphism. There is a variety of different types of polymorphism ranging from single nucleotide change to very large scale changes. For evolution and the Speciation such polymorphism play very important role, and you will study these in details at higher classes. The technique of DNA fingerprinting was initially developed by Alex Jeffreys. He used to satellite DNA as probe that shows very high degree of polymorphism. It was called as variable number of tandem repeats. The technique are used earlier improved southern blot hybridization using radio labeled BNTRs as a probe. It included isolation of DNA, digestion of DNA by restriction nuclease, separation of DNA fragments by electrophoresis, transferring, blotting or separating DNA fragments to the synthetic membranes such as nitrocellulose or nylon. Hybridization using labeled BNTR probe and detection of hybridized DNA fragments by autoradiography and a schematic representation of DNA fingerprinting is shown. The VNTR belongs to a class of satellite DNA referred to as a mini satellite. A small DNA sequence is arranged randomly in many copy numbers. The copy numbers varies from chromosome to chromosome in an individual. The numbers of repeats show very high degree of polymorphism. As a result, the size of the BNTR varies in size from 0.1 to 20 kb. Consequently, after hybridization with BNTR probe, the autoradiography gives many bands of differing sizes. These bands give a characteristic pattern of an individual DNA. It differs from individual to individual in a population except in the case of monozygotic twins. The sensitivity of the techniques has been increased by use of polymorphism chain PCR. You will study about in chapter 9. Consequently, DNA from a single cells is enough to perform DNA fingerprinting analysis. In addition to the application in forensic science, it has much wider applications such as in determining population and genetic diversities. Currently, many different probes are used to generate DNA fingerprints. So that's it. Thank you so much.